Hello, and welcome to this lecture on the derivatives of the three germ layers. What we'll be looking at is the formation of each of the following: the ectoderm, the mesoderm, and the endoderm. We'll look briefly at tissue signalling between the different layers that gives rise to different structures of the body, and some interactions between tissues themselves. And finally, we'll note that this has some implications for cancer. So let's go back to the beginning, to the stage of gastrulation, when the main body axis is formed. It has been described as the most important thing that ever happens to you. And in our diagram, in the upper image, we have the amniotic cavity marked in green, and beneath that the yolk sac marked in yellow. And between the two of them is a bilaminar disc made up of ectoderm on the top and endoderm in the bottom. And if you were inside the amniotic cavity looking down, in the floor, you would see the primitive streak start to form. So you can see that marked A and B in the diagram, round about day 16 after fertilization. And this is a key moment. It's the moment of individuation. So from now on, you could no longer be twins in any form. It also determines the main body axis, so it establishes left and right, and head is towards the center of the disc, and tail is towards the margin of the disc. But also, crucially for our purposes today, it leads to the formation of a third germ layer in between the ectoderm and endoderm. And that third layer is the mesoderm. And what will happen is that the ectoderm on the top surface will begin to migrate towards the midline. So moving towards the middle. And if we saw that in section, where we have the ectoderm or epiblast on the top and the endoderm in the bottom, as the cells move towards the midline, so as they reach that midline, they form bottle cells, they change their shape, and then they detach and fall off, and then they will migrate laterally out to the sides. So forming that third mesodermal layer in between the ectoderm and endoderm, a bit like the filling in a sandwich. So spreading out from either side to give rise to the mesoderm. That primitive mesoderm we'll find give rise to a variety of other tissues subsequently. Now, I've represented it by a schematic diagram with ectoderm, mesoderm and endoderm represented in these simple ways. And we'll use that consistently. But if we were to relate that to an image of an embryo, here we can see the amniotic cavity shown in yellow, and then the ectoderm shown in blue, the mesoderm shown in red, and the endoderm shown in yellow. And that endoderm, of course, is lining the yolk sac. There's also a cord-like structure which runs down the length of the embryo, and that's called the notochord, and that's present in all vertebrates, although it will largely disappear later on. So here's the notochord seen in between the two leaflets of mesoderm on either side. And that notochord, we'll find, has an important signalling purpose. So, in other lectures, we've looked at the formation of the neural tube from the ectoderm, and we'll touch on that again briefly here. But I've represented that as an ectodermal derivative lying underneath the ectoderm and just above the notochord. And that neural tube will later form the spinal cord in the adult. And here's an image of how it's formed. The tissue on either side, the ectodermal tissue on either side, will roll up and meet to fuse and form the spinal cord. And the tissue at the very margins gives rise to another tissue called neural crest, which is important in itself and will give rise to a number of interesting and different derivatives. Now, the mesoderm will also begin to segment into different areas. So moving out from the center, on either side, there'll be little cubes of tissue, of mesodermal tissue, called somites. Then there'll be a patch of intermediate mesoderm, and then lateral mesoderm out to the sides from that. So the mesoderm begins to specialize out from the center, out to the sides, into different kinds of mesoderm. And these will give rise to different derivatives later on in the course of development. In this image, we can see a picture of an actual embryo seen in a scanning electron micrograph. And up at the top, we can see the brain. Running down the center is the spinal cord, and on either side are the segmented structures which represent the somites. The sac lying underneath the embryo is the yolk sac. So that gives you an idea of what this looks like in the three-dimensional form of a real embryo. So back to our diagrams again. And in this one, I've added some arrows to show some of the signaling that takes place. So for instance, the ectoderm is signaling to the upper part of the neural tube, and the endoderm is signaling to the lower part of the neural tube. The endodermal signaling will help turn the lower part of the neural tube into motor nerves, and the ectodermal signaling will help turn the upper part of the neural tube into sensory nerves. And these will migrate out from the neural tube through the rest of the body. However, the neural tube itself also signals. It will signal to the somites, and the somites will begin to differentiate, as we'll see, into different regions which give rise to different parts of the body. Similarly, the endoderm is signaling to the lateral mesoderm, and that interaction will help form blood cells. The lateral mesoderm, in turn, is signaling to the ectoderm, and as we'll see, that will help provide all of the different forms of ectodermal covering that we see on the surface of the body. So structures like hair and teeth, etc., are all things derived from the ectoderm under the influence of signals from the mesoderm. In this picture, we can see neural crest cells at the very tip, the very apices of the parts of the neural tube that are growing together. And when the neural tube fuses, then the neural crest cells are isolated and left separate. And there they give rise to a variety of different kinds of cells. They will migrate individually throughout the body and give rise to pigment cells. So the cells that indicate skin colour derive largely from neural crest cells. But they also give rise to nerve cells, autonomic nervous cells, and sensory nervous system cells. And in addition, and very crucially to nerve function, they will give rise to the supporting cells of the nervous system. For instance, the Schwann cells, which wrap round the nerve fibres themselves. However, perhaps unexpectedly, they'll also give rise to cells which are like mesoderm, even though neural crest is originally derived from ectoderm. And in fact, they can also give rise to endocrine-secreting cells as well. So a very wide variety of cells, all coming from neural crest, rather unexpected in terms of the range of differentiation states that you can get from cells of a single origin of this kind. So, looking at the ectoderm and the epidermis, we already indicated that the neural tube will give rise to the brain and the nervous system. But in combination with the mesodermal signals, will also give rise to a variety of derivatives that are visible on the surface. So, for instance, tooth development is initiated by a signal from the mesoderm in a particular place, and that induces the overlying ectoderm to begin to form teeth. Similarly, hair is induced by the underlying mesoderm, but begins as an ectodermal growth. Incidentally, it's the mesoderm that determines which forms. So a piece of ectoderm from almost anywhere in the embryo could be made to form teeth or hair, depending on the signal from the underlying mesoderm. Sweat glands and salivary glands are also developed in the same way, and mammary glands, which are also secretory in function, are also initiated by the signals from the underlying mesoderm, talking to the ectoderm lying above it, and giving rise to the development of the mammary glands. Of course, these form identically in early stages in both males and females. It's female sexual signals which will subsequently give rise to the full development of the mammary gland in the female, but not so in the male. And the final, and perhaps the strangest of tissues that develop from the ectoderm, is the transparent cornea. Your eye is actually covered by a layer of ectoderm, but it's transparent at that point, so that you can see light signals passing through the cornea to reach the retina. And again, that transparency could be induced in any ectoderm with the appropriate signals from the underlying mesoderm. There are also two special senses structures which arise from the ectoderm, and those are the placodes that will form the eye itself. They will thicken and form an indentation to finally form the lens of the eye, and the ear placode, another thickening which will sink under the surface and form the inner ear apparatus, the semicircular canals and the cochlea. Now let's look at the mesoderm in our same schematic diagram. And I've arranged it in the same way, except up above, I've placed the heart mesoderm, because if we were looking down on the embryo from above, heart mesoderm actually starts out in front of the head. It starts out above where the head is going to be, and it's the folding of the embryo which brings the heart mesoderm underneath the head. And we will look at folding of the embryo in another lecture. We've already mentioned the somites, but so much happens with the somites that we'll have to give some special attention to those. At this point, we'll merely comment that the vertebrae and some striated muscle cells migrate out from the somites. The intermediate mesoderm will give rise to kidney, and the lateral mesoderm will give rise to blood vessel linings, the endothelial cells, to the cartilage, which will eventually be replaced by bone, and by the smooth muscle in the body. Also contributes to the mesoderm, underlying the skin itself, forming the dermal layer. Now, I mentioned that the somites are particularly complex and interesting, and here we can see a suggestion of that complexity. So, looking down from the top, the dermatome is going to contribute to the dermis of the skin. The myotome will give rise to all of the striated skeletal muscle cells of the body, so cells will migrate out into the body and give rise to all of the striated skeletal muscle. And then the sclerotome parts will begin to grow round the neural tube to form the vertebrae. This is a rather complex process, and in fact 4 somites will contribute to each single vertebra. But this is a general principle in development where if you wish to make a complicated bone, you often do it by fusing together a number of different elements. It seems hard to instruct a bone to form a very complex shape like a vertebra. So instead, what the body does is to build it up from a number of different components. Now let's have a look at the third layer, the endoderm, the lower layer of the 3. And this will form the linings of a number of other structures. So for instance, the gut, the lungs, the liver, the pancreas and the bladder are all lined with cells derived from the endoderm. But as these linings grow out, often in the form of buds, what happens is that mesoderm will condense round about them. So the final adult gut, adult lung, adult liver, and so on are composed of an endodermal lining, but with a mesodermal ground substance which fills up the bulk of the organ itself. If we look at that folding process in transverse section, so that's a cross section across the body of the embryo, in this image you can see the amniotic cavity at the top, and the lower part is the yolk sac. And as the folding of the embryo proceeds, the amniotic cavity sweeps round and begins to pinch off a small part of the yolk sac. And that small part being pinched off will form the gut tube. Perhaps easier to understand this in a view from the side. So here again we have the amniotic cavity at the top, the yolk sac at the bottom. You have to imagine the amniotic cavity sweeping round as the embryo rises up into the amniotic cavity. And the effect this has on the foregut and hindgut is to gradually enclose them as the gut sweeps round. And we've indicated that by shading in this particular image. So that indicates the fore and hindgut. However, as we indicated, there are also crucial interactions between tissues. Not everything in the body is specified in advance. And this is the explanation for much of the variation between individuals. No 2 people are exactly the same. And this is important in surgery, for instance, where things like the course of a nerve is predictable but not identical between different individuals. And therefore surgeons have to take particular care not to cut a nerve or interrupt a blood vessel when they're carrying out an operation procedure. And the reason for this variation is that many of these interactions between tissues are not specified in detail by the genome. Instead, what happens is that tissues are laid down in general positions and then they begin to talk to each other so that they'll establish a functional relationship over time. We can actually explore this in tissue culture. So this is a rather schematic diagram. As you can appreciate, the actual experiment is much more complicated and much more difficult. But here I've imagined placing a piece of muscle from an embryo into a culture medium, and alongside it I've supplied some nerve and some blood vessel. And what we find is that under the influence of factors from the muscle, nerves will be attracted towards it. For instance, there are a variety of nerve growth factors, but also blood vessels will be attracted towards it. There are a variety of what's called angiogenic factors, which can diffuse out from the muscle to interact with tissues on either side. So what will happen is that blood vessels will be attracted towards the muscle and nerves will be attracted towards the muscle. In fact, if we also had some cartilage present and some developing tendon, what we'd find is that there's a kind of seek and join rule for tendon and muscle. So the tendon will find the end of the muscle and attach itself to it. It will also attach itself at the other end to cartilage. So you can see that even in tissue culture environment, it would be possible to build up an almost kind of functional relationship between all of these different tissues. And this is particularly important when we come to look at cancer cells. Cancer cells have escaped from control of normal cell division, and they are increasing in number quite rapidly. But normally, as a result, what would happen is that they outrun their local nutrient supply, and they would form a little ball of cells, perhaps hollow in the center, because the cells in the middle had not had sufficient nutrients to survive, and they'd be dying and perhaps being replaced by pus. So you'd have a small ball of cells which was effectively going nowhere. However, if as a result of a mutation within the cancer cell, in addition to the mutations which made it divide rapidly, the tumour cells are producing an angiogenic factor, then blood vessels will be attracted towards it. And the tumour angiogenic factors are very similar to the angiogenic factors found in normal embryonic development. Once a blood vessel has reached the tumour cell, then the tumour can begin to grow rapidly. And if cells detach from the tumour and enter the blood system, then they can tumble around the body and begin to invade somewhere else. So the tumour can begin to spread elsewhere in the body as a result of the vascularization of the supply of blood to the tumour. And in some ways, this is one of the most serious steps in cancer development, because it both allows the cancer to grow and to spread. And you can understand that this is a major field of research interest, trying to explore the nature of these factors and how they might be prevented from acting to support tumour growth. So then, let's look at a summary of what we've talked about in this lecture. We looked at the derivatives of the germ layers, the ectoderm, the mesoderm, and the endoderm. We touched on tissue signalling between mesoderm and ectoderm, between ectoderm and mesoderm, and so on. And then we looked at the interactions between tissues, such as attracting nerves and attracting blood vessels towards developing structures. Finally, we saw that this has important implications and perhaps even some hope for cancer treatments. Thank you.